Hi, this is Amanda. You don't know her, but Amanda's my friend. She's a vegetarian, she exercises, she has never smoked. She's a yoga teacher, basically the kind of person who makes you feel bad about how you treat your own body. One day, she starts feeling strange. A cough that does not go away, shortness of breath, and expected she goes for a checkup. After imaging, biopsy, and genetic testing, the doctor finds the problem. Amanda has a non-small lung cancer, and her tumor carries an EGFR mutation. EGFR stands for Epidermal Growth Factor Receptor. The name sounds complicated, but simply EGFR is one of the molecular switches that tells cell when to grow. In healthy cells, this signal is controlled, but in Amanda's tumor cell, the EGFR gene has a small mutation, like a typo in the DNA instruction manual. The cancer cell build EGFR slightly different, and that small change can make EGFR overreactive, like a growth switch stuck on the on position. So the cell keeps receiving the message grow, divide, and survive repeatedly. Amanda's problem is not just a rare case. The World Health Organization reported about 2.5 million new lung cancer cases and 1.8 million deaths in just 2022, making lung cancer the leading cause of cancer death globally. Traditionally, cancer treatment focused on removing or destroying the tumor. Surgery, radiation, chemotherapy, these treatments are still important, but they are not always molecularly specific. Then cancer treatment moved toward targeted therapy, which instead of asking only where is the tumor, we ask what molecular defect is driving this tumor. For EGFR mutant lung cancer, the defect is overreactive EGFR signaling. So doctors can use EGFR inhibitors like osimertinib to block that abnormal activity. At first, Amanda's tumor may respond. The treatment works, but cancer is not a frozen target. Proteins are not statues. They move, they breathe, and shift between different shapes and conformations. A drug may bind well to one EGFR shape, but poorly to another. A resistant mutation can shift the binding pocket or change the protein energy landscape. So the real question is not just only, can we find a molecule that binds EGFR? It is actually, can we understand how EGFR changes during resistance and design that is aware of those changes? Our project is called MoquPilot. It is a hybrid AI quantum framework for exploring dynamic EGFR conformational landscapes related to drug resistance. The main idea is simple. MoquPilot does not treat EGFR as one fixed 3D structure. It treats EGFR as a moving molecular system that can pass through many possible conformational states. AI helps decide where to look first. Quantum-assisted optimization helps explore the reduced conformational state. And classical simulation helps check whether the predicted states are physically meaningful. The final output is not just one molecule, it is a resistance-aware map of EGFR shapes and candidate therapeutic strategies. The system has 4 layers. First, the biological input layer. The user-defined problem, EGFR mutation lung cancer, the mutation type, and the therapeutic goal. Second is a conformational analysis layer. AI analyzes protein structure, mutation data, known inhibitor interactions, and flexible regions. Its job is not to replace physics, but its job is to tell us where to focus. For example, it may suggest that the ATP binding pocket or key resistant residues need deeper modeling. Third is the quantum-assisted optimization layer. After the important region is selected, the conformational search is turned into an optimization problem. Possible protein shapes become computational states. An energy function rewards realistic low-energy conformations and penalizes impossible ones, like atomic overlap or broken geometry. Then, hybrid quantum classical method can explore this reduced state space. Fourth is the validation layer. Predicted conformations are checked by docking, molecular dynamics, ensemble comparison, and drug likeliness evaluation. So the logic is AI narrows the question, quantum helps search the reduced landscape, and classical simulation validates the answer. Our research has three main aims. The first aim is to build dynamic EGFR conformational model. Instead of using one static structure, MoquPilot generates an ensemble of normal, mutation-associated, and resistance-related EGFR state. The second aim is to identify resistance-associated states through hybrid AI quantum exploration. AI prioritizes biologically relevant regions, and quantum-assisted exploration investigates difficult parts of the reduced conformational landscape. The third aim is to evaluate candidates therapeutic across multiple EGFR conformations. Instead of testing a drug against only one perfect protein structure, MoquPilot asks whether the candidate can remain useful when the protein changes. That is the key point. Not just the best molecule for one structure, but the most robust strategy across dynamic resistance state. Now let's return to Amanda. Her tumor carries an EGFR mutation, and over time resistance may begin to emerge. In the current situation, we often understand resistance after treatment starts failing. MoquPilot explores a different direction. It tries to model how the target evolves, which conformations become important, and which therapeutic strategies may still work before resistance becomes dominant. So for Amanda, the future treatment could not be only personalized, but also adaptive. Of course, this is not easy. The first challenge is computational complexity. Dynamic protein modeling means exploring a huge conformational space, and quantum hardware is still limited. So if the quantum layer is not practical, plan B is to use classical enhanced sampling method, like accelerated molecular dynamics, metadynamics, Markov state modeling, or ensemble molecule dynamics. The second challenge is that full EGFR may be too large for quantum level treatment. So plan C is to reduce the system. Instead of modeling the entire protein, we focus only on the chemically important region, the ligand, the IATP binding pocket, and key resistance residues. The rest of the protein is treated classically. The third challenge is biological validity. Any predicted conformation must be checked by molecular dynamics, docking, comparison with known structures, and ensembles-based evaluation. So even if the full AI quantum version does not work perfectly, the project still gives useful outputs: dynamic EGFR ensembles, resistant-related states, and better candidate screening. If this project succeeds, the impact is not simply one better EGFR drug, but the bigger impact is a shift from static structure-based drug discovery to dynamic landscape-based drug discovery. Today, many workflows ask what molecule bind with protein structure, but MoquPilot asks what therapeutic strategy remains effective while the protein is changing. For biomedical engineering, this project connects molecular modeling, AI, quantum-assisted exploration, and therapeutic design into one adaptive framework. This is the vision of MoquPilot. Not just finding a drug that's for a static target, but designing therapy for a moving molecular enemy. Thank you for listening to us. This was a project for Biomedical Engineering class, GIST University.